This is going to be, or this is a new feature we're trying to introduce to the Net Science Center. I've been calling it a synchronous workshop. So one of us tutors are going to be recording some different workshops regarding to any of the classes we've been teaching in the Net Science Center, and these ones are going to be the ones that we consider as useful for you to have knowledge about. And then we're going to be uploading a video with the workshop in general, and then you can go over on your own pace. For this one, we're going to be covering a bio-10A topic, which is coming from Campbell's Biology Chapter 8 book, and it is called Metabolism. But again, if you find that this is going to be useful for you in any other of your biology class or in any other of your classes, just feel free to use it, and we'll be really glad that you're taking advantage of this. Anyways, metabolism. First of all, some key features you should be familiar with. If you already took the metabolism lecture, you should be familiar with these topics. So again, if you feel that this is kind of a review for you, just check it out that you actually know all these concepts. Okay, the first it says about organisms metabolism, and this is quite of the basis that and that metabolism transforms matter and energy. Then you should also be familiar with the idea of free energy and how can the free energy change tell us about the spontaneity of a reaction. Then we also have ATP and a little bit about the structure of ATP, and especially how ATP works on coupling exergonic and endergonic reactions, which is quite of the basis of metabolism. Then by the end, we're going to be talking about enzymes and how enzymes worked, how enzymes can speed up reactions by actually lowering the energy barrier. We're also going to be discussing about the active reaction energy a little bit, so you can be feeling familiar with that. And then by the end, you should also be familiar with the idea that the regulation of an enzyme activity can help actually the control of metabolism, which again is something really useful. And well, we're going to be going over that in detail in a minute. Anyways, first topic: metabolism transforms energy and matter. Metabolism in general is just a sum of chemical reactions of an organism. Okay, there could be thousands, there could be hundreds of reactions. There's not an actual limit. Okay, the energy, because we've been talking here about how this metabolism transforms energy and matter, but this energy generally is extracted from different sources that can actually store energy. Generally speaking, these ones are going to be complex molecules such as sugars or other fuels. Then, and what is a metabolic pathway? Well, generally speaking, it's quite of the same thing as metabolism, and it begins with a specific molecule and it ends with in a product. Okay, so that's kind of the basic idea of a metabolic pathway. And then each step of a metabolic pathway is catalyzed by a specific enzyme. This is something really important you should be understanding, because this is quite of the middle stones of the specificity of enzymes. They can be specific and they can control one reaction. So it kind of like helps a lot for the supply and demand, as you can see in the next bullet point. Okay, so that there's these ways to control the enzyme can actually help us to control the metabolic supply and demand. And finally, some pathways can have more than one starting molecule. This is also something you should be familiar with. Then this is in a small scheme that kind of like represents the way biologists portray a metabolic pathway. You can see there's a starting molecule and there's also a product. You have, in this case, a 3 steps a pathway. Each one is entails a reaction, and each reaction has it's a specific enzyme. There are 2 main metabolic pathways. The difference between one and the other is whether they use or they produce energy. In the case of an anabolic pathway, it actually consumes energy, and it consumes energy because they want to build complicated molecule from simpler ones. So they're kind of like making things more organized, which is something that we're also going to be covering in a second. Sometimes it is also called as a biosynthetic pathway, and an example of this one could be the synthesis of an amino acid from simpler molecules, and the synthesis of a protein from amino acids, which is kind of the thing you can see in the bottom of that rectangle. For a catabolic pathway, which is quite of the opposite, you have a group of reactions that releases energy, and this release is thanks to the breaking down of complex molecules into simpler compounds. The result of this is that we're going to be having energy available to do work, which is something that cells really want a lot. An example of this one is it could be cellular respiration, which is quite of the most common and widespread example. You can see we begin with a really complex molecule such as glucose, and after a series of steps, the cellular respiration yields ATP, which is a source of energy. Energy produced by a catabolic pathway can be stored and then used to carry on anabolic pathways. This is quite a really basic concept. It is one of the key concepts I highlighted at the beginning, and you should be really strong. You should be feeling strongly comfortable with this concept. Then what is bioenergetics? It's just the study of energy flow through living organisms. Forms of energy. We've been talking about energy for a while, so now we're going to be delving into energy in a little more detail. Energy is the ability to rearrange a collection of matter. I know that for many of you, the definition of energy is more the one that says that energy is the ability to do work. I rather include this one because it actually accounts for many of the biological moments or biological explanations in which we use energy. And again, this also actually accounts for the ability to do work, because the ability to do work is just referring to moving things around, which is kind of rearranging a collection of matter. Then there's two main categories of energy. There's kinetic energy and there's also potential energy. And then we have some subdivisions of that main energy sorts, which is thermal energy and potential and chemical energy. And well, we're gonna be going over those right now. First of all, kinetic energy. It's just the energy associated with relation of motion of the object. That's just an energy that is related to motion. Then we have thermal energy, which is a type of kinetic energy that is associated with the random motion of atoms and molecules. And then we also have heat, and heat is just the transferring of thermal energy. Potential energy is the other big guy, and in this case this is energy that matter possesses because of its location or its structure. And then chemical energy is a subdivision of potential energy, and this one is just one that is available for release in a chemical reaction. This is going to be a self-assessment station. I'm going to be asking you a question, then you can pause the video and try to see if you can answer the question on your own, and then I'm going to be giving you the answers. So the question says, how does energy relate to metabolism? You just can go ahead and pause the video and see if you can answer it. Well, what is the relation between energy and metabolism? Of course, and the basic one is that metabolism in a way produces energy, or either produces energy or consumes energy. But there's also a much profound connection, and it is that energy can actually control how fast or how slow, or with this idea of supply and demand, it also works as well. So how much of the products of a metabolic pathway we have. If we have more energy, you're kind of like telling metabolism that you do not want more, for example, catabolic pathways, because you already have enough energy. And vice versa, if you have many products, you kind of know that you're kind of like going out of energy, and it kind of relates in that way. Energy can actually control how metabolism behaves. Then the next topic is going to be about loss of energy and transformation. Thermodynamics, which is kind of the big thing here. Thermodynamics is the study of energy transformation. What is a system? What is a surrounding? What is an isolated system? A system is quite of a basic concept within thermodynamics, and it refers to the matter that is studied. If I'm studying the energy or the heat that some pencil releases, that pencil is going to be a system. Then what about a surrounding? A surrounding is just the rest of the universe. So the pencil is, in this case, in the example of the pencil, your pencil is your system, and the whole universe is the surroundings, which is kind of quite impressive. Then we have two concepts regarding systems. Your system could be isolated, or your system can be open. If a system is isolated, it means that there's no transferring of energy between the surrounding and the system. Whereas in the case of an open system, it's quite of the opposite. Now the surrounding and the system are transferring energy. It could be flowing out of the system, or it could be getting into the system. Okay, but it just means, an open system just means that there's some transfer of energy. The first law of thermodynamics, which is also referred as principle of conservation of energy. This is one of the pillars of thermodynamics, and I'm pretty sure you've heard of this before, even if you haven't taken the metabolism class before. Energy can be transferred and transformed, but it cannot be created or destroyed. This is quite an important concept. Energy is something that remains. It just changes forms. It could be created nor destroyed. Entropy. This is another important concept within thermodynamics. Entropy is quite a complicated topic, but this definition is quite simple to understand. Entropy is just a measure of molecular disorder and randomness. Then it comes, based on this concept, it comes the second law of thermodynamics, that it says that every energy transfer must increase the entropy of the universe. Okay, in other words, and again, there's something that I want to point out first of all for this law, and is that this law is specifically for spontaneous reactions. Okay, so it would be better worded as, for every spontaneous reaction, transfer of energy must increase the entropy of the universe. Okay, so this just applies for spontaneous reactions. Okay, because the transfer of energy could even happen when it's the opposite, that you should be inputting a lot of energy. Okay, and we're going to be talking about this idea of spontaneity later, but for now that's just what you should be familiar with. So the only thing the takeaway from here is just that the energy transfer creates this order in the universe. Okay, in a ecosystem, energy generally enters as light and is released as heat. Okay, this is something important to know. You can see here that the second law is it's being followed. Light enters as a really organized way, and when it's released, you can see that it's released in the form of heat, and heat is a really disorganized type of energy. Organisms create both organized products from disorganized materials and vice versa. This is something important, and as I tell you before, because something because some product is organized, it does not necessarily mean that that it actually do not increase the entropy of the universe. Sometimes creating organized things actually could further ensure the production of unorganized consequences, which is also quite crazy. Another self-assessment station. In this case, the question says, which metabolic pathway could you identify as creator of order, and what about the other metabolic pathway? So again, you just can stop the video and see if you can answer the question. Now, this is a quick recall from previous slides. There's two main sorts of metabolic pathways. You have anabolic and catabolic pathways. An anabolic pathway creates complex molecules from simpler ones, and this creation thing could be also referred as a creation of order. You are bringing order. You're taking simpler molecules into complex and more organized ones. Whereas in the case of a catabolic pathway, you're breaking things apart, so you are creating more disorder. Okay, so anabolic pathway is a process that organizes matter, and catabolic pathway is one that actually causes disorder. That was the question. Let's go ahead. Gives free energy and spontaneity. Spontaneous processes. I've been discussing about this for a while. Now you'll see what's the definition of an spontaneous processes. It happens without the input of energy and can be quick or slow. This is really important to know. So, if something is spontaneous, the only thing that tells you is that it did not require energy to happen. But it does not tell you anything about the rate of that process. It could be slow, it could be quick. For a processes to be spontaneous, the entropy must increase. Okay, again, it is one kind of like goes back or traces back to the second law of thermodynamics, as I stated when I was explaining that, when I was refreshing that. You can see here that for a process to be spontaneous, the entropy must increase. Free energy, or gives free energy, helps us determine if a process is spontaneous. What is free energy? Free energy, that is also that is represented with a big G, is the energy that can do work. Okay, when temperature and pressure are uniform. What does it mean is that certain reactions or certain processes have some associated energy, and that energy, or well, I don't know, like it's, for example, if you're decided to break a bond, that energy that is going to be released is going to be available to the work, and hence that is the free energy of that reaction. Okay, then how can we identify if the reaction will be spontaneous or non-spontaneous based on the free energy? If the free energy change that is represented as delta G is negative, which means that it's exergonic, and exergonic is just a fancy way to say that it releases energy. Okay, so if delta G is negative, the reaction will be spontaneous because it releases energy. And as I stated before, the release of energy is always something that increases the entropy of the universe, so will be spontaneous, and thus releases energy. If delta G is positive or zero, which means that it is endergonic, which is kind of the same fashion of the exergonic word, but in this case endergonic just means that it needs energy to be performed. And again, if this is positive, or if delta G is positive of zero, the reaction is going to be non-spontaneous. This is just a small chart that compares both endergonic and exergonic reactions. You can see that the difference between the reactants and the products is the delta G, and you can see that for the graph in the right, when the reaction is exergonic, delta G is negative because the reactants have more energy than the products, and on the other hand, for the endergonic reaction, is just the opposite. And you can see there's the 3 bullet points important that you should be taking away from these two concepts. Cells are open systems. I hope you remember the definition of an open system, and that's never at equilibrium. And this is such an important thing, because if something is at equilibrium, there's nothing happening in there. Okay, if you have a pencil that is at equilibrium, the pencil is not moving, so you have no way to carry on any process. But systems, but cells or systems are not at equilibrium. I will never be at equilibrium. Okay, a key feature of life is that metabolism is never at equilibrium. Okay, because this seek for equilibrium sometimes is the force that moves some reactions. Another self-systematization. How do living organisms could take advantage of spontaneity in some processes? I'm just gonna again just go ahead and pause the video and see if you can answer the question. Okay, how living things take advantage of spontaneity? As you may recall, spontaneity is something that does not require energy. Okay, And that's kind of, if you want to make a comparison, it's just kind of like going to a restaurant, eating everything you want, and then the owner of the restaurant just allow you to leave the restaurant without paying. And that's kind of the idea of spontaneity in these cases. So our cell can do some processes, and our cells do not have to pay for it. You can assume that the currency that our cells uses is energy, and processes that are spontaneous do not require energy. So our cells actually take a lot of advantage of this, because again, you do not have to make any extra process, you do not have to input any energy, it just will come, it will happen. And that's something and that's how our cells take advantage of this, because it actually do not require energy. And many of the biological reactions that happens within our bodies are actually spontaneous. ATP powers cellular work by coupling exergonic reactions to endergonic reactions. This is again another of the key concepts you should be familiar with. A cell does three main types of work. You have chemical work, that is just pushing endergonic reactions. Do you remember endergonic needs energy? So that's the type of chemical work. It's kind of like inputting energy for endergonic reactions to happen. Then transport work, which is just pumping substances against the direction of a spontaneous movement. And then you also have mechanical work, such as the contraction of muscle cells. Those are the three different types, and we're going to be going over each one. Energy coupling, cells' way to manage energy. This is a key concept that we're going to be covering now. And this energy coupling, as I just said, it also uses exergonic process to drive endergonic ones. This relation is quite of the most important ones within our cells. You know that exergonic processes releases energy, and endergonic processes need energy. So they kind of go along with each other, because the needs of the one are the products of the other, and our cells manage a way to couple them out. And the way we call that coupling is that energy coupling. And ATP acts as a mediator for energy coupling, and as intermediate source of energy to cellular work. The way exergonic and endergonic reactions are tied together is by means of ATP. And as I said before, ATP is also a source of energy. So the way exergonic reactions share their energy is basically by means of ATP. So they are kind of like sending ATP as a gift to endergonic reactions, and endergonic reactions are taking the ATP, and then they're using that for carrying on its processes. Now let's go to the structure and hydrolysis of ATP. ATP stands for adenosine triphosphate, and is used to make RNA, besides its role in energy coupling, so it is also an important part of the RNA. A structure, you can see the structure of ATP down there. There's the ATP is just the grouping of 3 phosphate groups, a ribose, which is a sugar, and adenine, which is an amine group. Okay, you can see the 3 phosphate groups, and there's something important that I want to highlight here. You can see that each one of the phosphates bears a negative charge on one of its oxygens. Okay, then we have the ribose, and we have the adenine. That's basically the structure of ATP. Let's go ahead and talk a little bit more about breaking things. The bonds between phosphate groups can be broken by hydrolysis. And hydrolysis just means a breaking by the addition of water. Okay, inorganic phosphate is released when this bond between phosphate groups are broken. And again, you can imagine, let's go back to the structure so you can see it. You can see that there's 3 different phosphate groups, so you can break the bond between one of the phosphates and the oxygen, and then you're going to have some phosphate being released, and that phosphate is going to be called an inorganic phosphate. And then when our ATP loses one of its phosphates, now it becomes ADP. T stands for tri, D for di. So it's going to be adenosine diphosphate. Now, this reaction is exergonic. You're breaking things, although this is not actually the actual source of energy, but you kind of have the idea that this reaction releases energy. Okay, but this is something important you should be taking away, or you should be familiar with this concept, is that this energy is not coming from the bond breaking. Okay, the phosphate bonds are pretty weak and they do not actually contain a lot of energy, and this is not really the way or where the cells actually take the energy from. And the actual difference between one and the other, or the energy, or where this energy is going to be structured, is because of the difference between the structure of ATP and ADP. ATP is a molecule that is highly energetic and it is in a really high state of energy, whereas ADP is way less energetic. And that change and that difference between the reactants energy and the product's energy is where this energy is coming from. Then you can see here's a small representation of the hydrolysis of ATP. You can see how water is entering and then one inorganic phosphate is being released. Self-assessment station. ATP is suitable because it yields generally more energy than other reactions within the cell. Why is that? I'm going to be giving you for this one a hint. You can just go ahead and check it out, the ATP structure, and see if there's anything you can find there that could be the reason why this ATP is so suitable. Just go ahead and stop the video and see if you can find it out. Well, let's go over the question. Why ATP produces such a great amount of energy? If you went over this structure, you noticed something that I also highlighted when I was explaining the structure, and it is that the ATP as a molecule bears three negative charges. Okay, and all of them are really close to each other. That makes ATP a really unstable molecule. And being such unstable implies that that molecule will have more energy. But when one of these phosphates is lost, you can actually, the two phosphate groups that are going to be remaining are going to be able to be more apart from each other, and that actually causes that ADP to be way more stable. And that is actually quite a big difference, and that's something that just happens with ATP, although our cell also uses other compounds as sources of energy. But that's why ATP is so suitable, and that's why ATP releases that amount of energy. So now let's go ahead and see how the hydrolysis of ATP performs work. In this case, we're going to be focusing in chemical work. Okay. And in this case, this is based on the usage of enzymes. ATP binds with endoorganic reactions with a delta G less than the delta G of hydrolysis of ATP. What does it mean is that as ATP binds with endoorganic reactions, okay, the reactions that are going to be using this ATP might be the ones whose activation energy, or this delta G, is less than the energy of hydrolysis, which kind of makes sense because if a reaction actually needs more energy than the one that we can produce, the reaction is just not going to happen. It's not going to be ATP is not going to be used in those type of reactions, and that's kind of the way it works. And we're using here enzymes. First of all, the first time we're introducing that word, and in this case, the idea of using enzymes for this one is that ATP sometimes binds, for example, to the enzyme, and these enzymes are also going to be helping us out to actually regulate how much energy the reaction will be needing to start. Okay, this happens by means of phosphorylation of the reactant. The phosphorylation is just a process of transferring of phosphate groups. I do have a diagram that shows it better, so just hang in there that concept. The new reactant, the one that now contains the phosphate group, is called the phosphorylated intermediate. You can see here there's an example that actually shows one of these processes. In this case, this one describes the how glutamic acid becomes glutamine. Okay, you can see that at the beginning, the reaction by itself, when you are mixing, let's say, glutamic acid and ammonia to produce glutamine, the delta G is actually positive, which means that it's not going to be spontaneous, which means that it will be needing a lot of energy to happen. Okay, but we do not want that to be the case. So now we're going to be introducing ATP. So now you can see in the step B how glutamic acid now is first binded to one of the phosphate groups from the ATP, creating a phosphorylated intermediate. Then ammonia is coming into the reaction, and then you can see how glutamine is created. Okay, and you can see in the last step overall that if you decided to calculate the net delta G, now it's negative, which again, we're kind of converting an endergonic reaction into an exergonic one. And that is actually the main goal of ATP. We want reactions to happen spontaneously, and ATP can make it happen that way. How the hydrolysis ATP performs work now in transport and mechanical work. Let's go over transport first. Again, this happens by means of phosphorylated intermediate, and the main key here is that when ATP binds to the transport protein, which is how transport work is done, the shape of the protein is changed. Okay, and this change leads to a change also in the binding ability of the protein. You can see in the example down there, when the protein is the one with a purple color, how when it is binded to a phosphate, which is that little p in the bottom of that image, you can see that that binding causes that this protein that at the beginning was able to bind to the solute, which is those balls in orange, it seems kind of like light orange. The thing is that now when this phosphate binds, it actually did not allow anymore this solute to be able to bind. So phosphate is binding, which means that the shape of the protein is changing, and if something changes its shape, it's not going to be able to bind anymore, and then the solute is going to be released. So this kind of take advantage of this idea that the binding is entirely dependent on the shape of the protein. Then we have mechanical work, which kind of uses the same concept. Now the difference is that ATP binds to a motor protein, which is those type of red legs that you can see down there, and the ATP motor protein bond is hydrolyzed. You can see that the ATP and the motor protein are actually binded, and we use our cells use hydrolysis to break that bond. And again, it's the same idea. You're breaking a bond, and since you're breaking a bond, the ability to bind of those red legs are going to be changing. And now the motor protein is going to be kind of given the impression that it is walking. Okay, and that's how this mechanical work actually happens. Now let's go over regeneration of ATP, which is also called ATP cycle. ATP cycle is the actual thing that binds endergonic and exergonic reactions. Okay, why? Because the whole cycle applies for the creation and the consumption of ATP, and in the part of the creation of the ATP, that's the part when exergonic reactions take charge on. And when ATP is used, now it's a turn of endergonic reactions, and that's how both are related. Couple things to know: ATP is a renewable source of energy. The process involves the addition of an inorganic phosphate to ADP, which is kind of like going backwards from hydrolysis, and then the energy comes from exergonic breakdown reactions. So this energy is coming from other exergonic reactions. And this one's is kind of the basics on how both things are actually connected. Now let's go over the topic of enzymes, which is going to be our last one. Enzymes speed up metabolic reactions by lowering energy barriers. What is an enzyme? First of all, an enzyme is a macromolecule that serves as a catalyst and is not consumed in the reaction. Okay, I hope you're familiar with the word catalyst. This is something that speeds up something. Okay, a catalyst is something that speeds up a reaction. They could be a protein, and the ones that we're going to be talking now for this workshop are going to be actually only proteins, but it's just good to know that there could be also RNA, okay, and those are called ribozymes. Many biochemical processes are considerably small, and that's why enzymes are so useful, because they can speed up these slow reactions. Activation energy barrier is the other topic we should be familiar with. Reaction involves bond breaking and bond forming. So when you're imagining a reaction, you can imagine that there's a compound, and that compound has certain bonds, and those bonds are going to be broken, and now new bonds are going to be formed, and hence a new compound is going to be created. When a reaction occurs, it goes to a higher unstable energy state. The way reaction works is that it begins with reactants, then it goes energy-wise, it goes all the way up until a point when the bonds are almost about to break, and at the same time they're also about to be created. Okay, and that state is really energetic and it's really unstable. The energy associated with that state is called the free energy of activation, or just activation energy. This energy is the energy needed for the reaction to start. Okay, so for a reaction to happen, the reaction should overcome this unstable state, and the way that happens is by introducing energy into the system. Then here's some small diagram showing you the energy diagram of a exergonic reaction. You can see that that EA in that kind of a hill, when it says transition state, it refers to the activation energy, and you can see that's quite of a big hill, and for that to be overcome you should be inputting energy, and that's kind of the way it worked. And you can see here this reaction is going to be exergonic because delta G is less than 0. Enzymes speed up the reaction by lowering activation energy, and the enzymes are specific for the reaction, and this specificity is going to explain it later, but just kind of like, you know, an advance of that concept. It is because of the shape of enzymes, and I hope you remember that proteins, that the ability of binding of things, as I explained before, it's quite related to the shape of could be the protein or the enzyme, which could be also proteins. I cannot I cannot make a reaction exergonic by means of an enzyme. Okay, we see it with ATP that it can actually convert a reaction into something exergonic, but in this case for enzymes it cannot happen that thing. The only thing that enzyme does is that it enhances reaction that will be happening anyway. This is a enzyme enhanced reaction. You can see how the activation energy is lowered for the red line, which is the one that accounts for the enzyme, and then you can see the black line that has a quite greater hill to be overcome, and that is the reaction that do not contain any enzyme. Enzyme specificity, as I told you, we're going to be covering this idea of enzymes being specific for a specific reaction. What is a substrate? That's kind of a basic concept, so we should be going over for this part. A substrate is a reactant at which the enzyme acts on. Okay, so this substrate is kind of the reactant of the reaction in which the enzyme can work on. An enzyme substrate complex. The way enzymes enhances processes, generally speaking, is by binding it to the substrate. So the enzyme goes and binds to the substrate, and it creates certain conditions that enhances the reaction. And this complex made by the enzyme and the substrate is called enzyme substrate complex. The active site The active site is at the region at which the substrate binds to the enzyme, and that's also the site where catalysis worked or happened. The specificity is attained due to the shape, as I just said before, and there's another concept you should kind of be familiar with, this idea of that an induced fit brings chemical groups to the active site into positions that enhances their ability to catalyze the chemical reaction. Catalysis in the active site. In catalytic reactions, the substrates bind to the specific site by weak covalent bonds. That's what I was explaining before, and how this catalysis of reactions happen. Some substrates are coming to the specific enzyme that, due to its shape, is able to bind with that substrate, and that binding is by means of really weak covalent bonds. Many metabolic reactions are reversible, and enzymes can enhance either of the reactions. That is something important, so you could enhance the forward or the reverse reaction. There's no preference. There's different ways enzymes lower activation energy. First of all is orienting substrates correctly. The idea of a reaction, as you may recall, is with bond breaking and bond creation. And sometimes the way our compound or our reactant is oriented into the space actually makes a big difference. Okay, the way I generally see it, it's kind of those little kid games when you have to place the correct shape into the correct hole. The circle goes into the circle hole, and so on. And this is kind of the same idea. So the way enzymes work is that it actually helps out to get into the right shape or into the right position, and it actually enhances a lot, and it actually reduces a lot also the activation energy. It also strains the substrate bonds. It kind of like stretches the bonds, and it makes it easier for them to be broken, and again it actually lowers activation energy. It also provides a favorable environment. Some reactions just happen under certain pH conditions, or with certain humidity, or perhaps temperature, and these enzymes can create this microenvironment that can actually enhance the reaction by lowering the activation energy. And finally, just covalently bonding to the substrate itself. This is kind of the catalytic cycle. You can see how it works. You have the enzyme at the beginning with a certain shape, then you have the substrates coming into the enzyme and binding with it. Then you have your complex, and then because of the position, the reaction is way more likely to happen. The reaction will happen, and then these products are going to be released. And something to be noticeable here is that for step number 3, you can see how the shape of the protein has changed because of the binding of the substrates. Self-assessment station. Again, another one which it's been quite a long time since the last one. How could concentration of enzymes could affect the rate of a reaction? A rate of a reaction is just referred to how slow or fast a reaction occurs. So again, just pause the video and see if you can answer it. Perfect. Let's go ahead and see the answer. How could you think that enzymes concentration could change the reaction? Well, if you have more enzymes, that implies that the substrates that are floating around are going to be more likely to bind to an enzyme, and therefore more product is going to be produced in some specific time span. Okay, so if I have more enzymes, I am going to be able to enhance the speed of the reaction. So the reaction is going to be going to happen faster. Whereas if I decided to lower the concentration, I'm going to have all these substrates floating around, but there's going to be just some minute and reduced spaces where they are going to be able to bind. Therefore, I'm going to be producing less products into the same time span. Therefore, the reaction is going to be slow. So how the concentration affects it? It could actually, if the concentration is higher, the reaction will go faster. If the concentration is lower, the reaction is going to be slow. Local conditions affecting enzymes. I I haven't discussed this yet, but you can see it now. Each enzyme has a certain pH and temperature that favors the most active form. Again, it just entirely depends on the shape of the of our enzyme in this case. If you have certain pH and temperature, our enzyme is going to be feeling more inclined to attain certain shape, and that shape is always going to be the most active one. Okay, certain chemicals can also affect enzymes, so it could actually even stop the working of an enzyme. We have here the two new concepts, cofactors and a coenzyme inhibitors. A non-protein enzyme helpers, that's what a cofactor is. It is also known as coenzyme when it is organic, and it can be binded permanently or not to our enzyme. The way it works, I'm going to be showing you. There's a graph that talks about it, and it's going to be in the next, I hope, the next slide. Then we have another concept here, which are the competitive inhibitors and the non-competitive inhibitors. A competitive one is the one that binds exactly to the active site. So now our substrate is not going to be able to bind there because there's something already there. Okay, and in the opposite case, conversely, a non-competitive inhibitor is something that binds on every other place within the enzyme but the active site. And the way it inhibits the process is basically because, again, if something binds to another place, it will change the shape of our enzyme. And as I've been remarking a lot along this workshop, the shape is such an important thing for the binding ability of an enzyme. Then we have this last concept when we're going to be seeing this idea of a cofactor as well. This is called an allostatic regulation, and that just refers to something that binds to an enzyme that enhances the ability of the enzyme to do its job. Okay, you can see in the allostatic activators and inhibitors, a part in the graph on the left, that in the case of an activator, it binds to some place and it stabilizes the active form. Okay, when it stabilizes it, it makes it more suitable or more likely for the substrates to bind there, and therefore speeding up the reaction. And then we also have inhibitors, and when they bind to some places, they are going to stabilize the inactive form, making it more complicated for the actual substrates to bind to the place, because now the shape that is more likely to bind with them is going to be disturbed. Then we have something that is called a cooperative regulation, which again, it just comes with the idea that a substrate will be binding to a specific place. Well, the thing is that when the first substrate will bind to one of these spots within our enzyme, the very idea of this thing bonding into that enzyme will stabilize the active form, allowing more of its counterparts to actually bind to the same enzyme. Something also to take into account for this concept of allostatic regulation is that these ones are accounting for enzymes that has more than one active site. Well, you can see that for these examples there are 4 active sites. And that's pretty much it all about this workshop for today. I hope you enjoy it, and most important, you actually find it useful. If you have any questions, you can always come to us to our office hours and go over some of the concepts or even some of the self-assessment questions if you find that those weren't actually clear, or if you just want more information about them. Here by the end, there's a check station. There are 2 quizzes I posted in there. They're going to be in the slides, so you just can open the slides and go over those. The practice test is quite an engaging one. It actually contains about, I think it's 50 questions, and that's something that you can, and that actually resembles a lot to what you can be finding for a test or for a quiz. Okay, again, it could take an hour or so, but if you can actually do that test, that means that you actually have a really strong knowledge about the topic. And then if you do not have that time, or you just do not going to go over that type of test, there's also the self-quiz, which is about 5-6 questions. It also covers the main ideas about the same idea of metabolism. Again, you can go over both if you want. And again, if you have any questions regarding these quizzes and these assessments, you just can go to our hours, to our office hours, to our tutoring hours to ask help for those as well. And then at the very end, there are our tutoring hours. From Monday to Friday, you just can make an appointment with us at any time. It could be even 1 minute after the actual appointment time, and we are going to be always pretty grateful to be helping you out. And that should be pretty much it. So